fractions between 0.3 to 0.8 molar. If you convert them in terms of a percentage, then it is 1.7 to 4.8%. Normally, if you go to this type of, the main source of this organism is seawater for a moderate halophilic organisms, because the salt concentration in a seawater is ranging from 0.6 molar to 3.5%. So that is very much similar to the slight halophiles. Then those organisms which are growing slightly higher concentrations of a salt, they are known as a moderate halophiles. So these organisms are growing from 0.8 to 3.4 molar of a salt concentrations, which is 4.7 to 20%. And extreme halophiles, which optimum salt concentration is above 20%, so which is range between 20 to 30%. These organisms will require sodium salts for their growth, and as we have discussed in case of a thermophiles and psychrophiles, the organisms which are not requiring those higher temperature or lower temperature but which are surviving. So similar way in case of salt also, a one group of the organisms which won't require higher temperatures, higher salt concentrations, but if you increase the salt concentration, they will not die. They are known as halotolerant organisms. So for your exam preparations, these two words are very important, because many times, tolerant and philic or tolerant and resistance, when this type of the terminology will come, then the temperature or the salt concentrations, the range will also go up or down, and accordingly you have to answer. Wherever it is a tolerant or means they are just surviving, it is not multiplying. So when you say that organisms which is surviving at 55 degrees centigrade temperature, but optimum temperature of this organism is 30, how you classify? Then they are not thermophiles. Or organisms which are surviving at 75, but which grow better at around 30 to 35 or 40 degree, then they are thermotolerant organisms. So this type of the answer you have to give. When it is asked that, if the organisms are surviving at up to 120 degree, but optimum temperature is above 80, then it is extremely thermophilic organisms. Similar way, when the concentration of salt is more than 20%, then it is a extreme halophiles. So accordingly we have to give the answers. Now, as we have seen in case of the temperatures, there is some change in the protein, then there is a formation of either the disulfide bonds or hydrogen bonds or some salt bridge. Similar way in case of this halophilic organism also, there is some change in the protein of these organisms, and they are known as a hydrophilic and hydrophobic. So organisms which are growing at a higher salt concentrations, we will see in some other factor which are affecting that water activity. So when you are dissolving higher salt or when you are dissolving higher sugar concentrations, it means you are reducing the water level. So this molecule, this organism, they are having the hydrophobicity, and due to this hydrophobicity they are able to survive, and particularly they are having the under reparations of a cis gene. So these are the two mechanisms by which these organisms, they can adjust their cell wall, they can make their protein, they can make their nuclear material to tolerate or to resist the concentrations. It is also evident that protein is less hydrophobic, particularly the name is given DHFR, that was found to have a narrow beta strand. At the DNA level, the halophiles exhibit distinct dinucleotides and the codon uses, and due to this, these organisms, they will be resistant to this. Now, temperature, salt concentrations, other is the pH or the hydrogen ion concentration and OH ion concentrations. So normally, many of us know when you take lot of oily food or farsan, then after 2 or 3 days you will get acidity. It means whatever the food has gone, it has increased, it has changed the slight pH, and due to this, you are feeling uncomfortable. While in case of this bacteria, there is a very, very broad range of a pH or hydrogen ion concentrations where they are growing. And depending upon either it is a very high concentration of hydrogen ion or less concentration of hydrogen ion and more concentration of OH ion, they would be classified or grouped as a acidophilic organisms, or we can say alkalophilic organisms. So acidophilic organism, if we want to go for a general definitions, then any group of organisms which are growing below pH 3, preferably below pH 2, then they are classified as acidophiles. Those organisms which are growing below pH 5, they cannot be classified as acidophiles. They may be acid tolerant, or they may be optimum pH may be pH 5, but for classification or grouping of the organism as a acidophiles, it should grow below pH 3, and that should be the optimum pH. Not that these organisms are growing better at 5 pH and they are growing some growth at a pH 3. If you take the example of Aspergillus niger, now it grows best at 5 pH, but it is not inhibited at 3 pH. So we cannot classify Aspergillus niger as a acidophiles. But a group of organisms which grow or which love the pH 3 and below 3, they are known as a acidophiles. We are having the organisms which is growing at 0.4 pH, extreme acidophiles. Acidithiobacillus ferrooxidans is one of the organisms. This organism you can find at a mine site, sulfidic mine, and due to the mine drainage, the pH of this water is normally 2 or below 2, and you will find this type of organisms. Acetobacter aceti have an acidified cytoplasm which forces nearly all the protein in the genome to evolve acid stability. Now, why I have given these examples? In case of the Acidithiobacillus ferrooxidans and other acidophiles, if you look to the pH of a cytoplasm, then it is as normal as our cell or as normal as other organism, and which is around 7. But in case of this Acetobacter aceti, the cytoplasm pH itself is acidic. And this particular organism is very important to know, because in case of other acidophiles, the cytoplasmic pH is around neutral. So the protein is not affected due to the acidity outside the cell. But in case of this organism, cytoplasmic pH is also acidic. So under this condition, if the protein and nuclear material is not deactivated or denatured, then what could be the reason? So these type of the extremophilic organisms, either psychrophiles, thermophiles, or halophiles, or acidophiles, now they are very important to know that by chance, now the atmosphere will change and temperature will go as it is increasing. No, every year temperature is increasing by 0.1 or 0.2 degrees centigrade. So suppose we think that you are going for another 200 years to survive, and at that time the temperature is 52 degree. What will happen to our protein? Can we survive? Normally, if the temperature will go 48 or 50 degree, many people will die. But if the scientists will work on this acidophilic organism or thermophilic organisms, and if they find out some particular gene or a protein, and if it will be transferred in a other organism, then this possibility will come that they will survive. In most acid-stable protein, such as a pepsin and SOX protein from Sulfolobus acidophilus or acidodurans, why this organism is having a particular different things. It is growing at a acidic pH. At the same time, now this Sulfolobus is also thermophilic organisms. So it is having two different extreme conditions, a temperature and pH. And what would be the combined effect of higher temperature and higher pH? So this, if you will study this, you will find out how the building block of these organisms, they are arranged, what are the binding force of this building block of these organisms, and they are not deactivated at the higher temperatures. Opposite to this acidophilic organisms, alkalophilic organisms. So similar way, this group of organisms which are growing in alkaline conditions. Normally, when we want to kill the organisms, the best, easiest available material is using soap. Detergent we are using, because that detergent is having the pH around 8.5 to 9. And due to this alkaline pH, the organism cell wall will get disturbed and organism dies. But in case of the alkalophilic organism, it is not at 8.5 pH or 9 pH. But some of the organisms, they are growing at 10 pH or 8 to 9 to 10 or 8.5 to 11 is the range of this, and 10 is the optimum pH for growth of these organisms. So the way we have discussed in case of a psychrophiles, thermophiles, asterophiles, so what is the change in the cell wall or cell membrane of this organism? So normally organisms, they will be get we can say lice at a alkaline pH. No, this is when you are using molecular biology, when you don't get anything, we use the detergent to lyse the cell. And so these organisms, if you are working with a alcoholic organism, they may not lyse by using the just detergent, and this will give you help to find out. Now the water activity, as we know this, we all require water more than any living cells. Then it is having more than 80% water, preferably around 90% water or 95% water. And what is the effect of water activity on the growth of microorganisms? If we look to the day-to-day use, then we know that when you are preparing pickles, how you are preparing pickles? You are adding high amount of salt. If you are preparing jam, then you are adding high amount of sugar. If you are preserving, suppose rice or wheat, now we are drying them, and as they dry. So all this activity, either you add some of the soluble compounds or you heat them, it means you are decreasing the water available in the cell. And when you are decreasing the water available in the cell, no, they get dehydrated. And once they get dehydrated material, then organism will not grow on this, because organism also require some water activity, and below this water activity, so it's a method of preservation. We can say the easiest way of preservations, you can decrease the water activity of any substrates or any material, then they are resistant to the microbial attack. But unfortunately, some of the organisms, now they are surviving at a low water activity. Many examples are there, fungus cultures and some of the bacteria. Normal organisms, the optimum water activity is around more than 0.9, while some of this fungi and some of this bacteria, the water activity would be as low as 0.6. Now, if you compare 0.9 and 0.6, now you will find it is a difference of 0.3, but you have to think in a different way. Now, in terms of a percentage or in terms of a fold decrease, even the water activity will go below 0.9 to 0.85. Now, normally we feel when we have the vomiting and the loose motion, it means your body is losing. Immediately, now person should be admitted into the hospital and the normal saline would be given. So, because to maintain the water activity. So, anything which is going below 0.85, it's very critical, very critical. And some of these organisms, they are growing at 0.6 water activity. And due to this, whatever the harmful effect what we get, many times, when you prepare a pickle, when you are adding high amount of salt, but suppose you are preparing this summer a mango pickle, and next summer or just in the month of January, February, you will find that pickles is slightly blackish in color, or the wall of the mango pieces, they become soft. Those who are not knowing, they will consider this the best way of preparing the pickles. But it is not. It is reverse. Whatever the structure of your mango or any pickles, it should remain as it is. If it becomes soft, it means it has been attacked by microorganisms. And when you are adding oil, when you are adding salt, or in case of jam, you are adding the sugar, you are decreasing the water activity. In spite of this, some of the organisms, those who are growing at a lower water activity, they survive and they multiply. And they attack whatever the lignin or cellulose or chitin which is present in the cell wall of whatever the pickles you have prepared. And due to this, the cell wall becomes mild or loose, and that's why it becomes this. So, now pressure. Normally, if you go to sauna bath or something, you feel some pressure in bathroom. And But what is the maximum pressure you can resist? Can you tell me? No, then because we are discussing about in terms of a activity of organisms. Normally 1 atmospheric pressures we all are more comfortable. It goes slightly above, then we feel slightly uncomfortable. And those who are knowing swimming, they know that if it is 10 feet, 20 feet, you can jump, you can dive, you can go up to the bottom, you can come back. But if it is more than 20, 30, 50 feet, then you have to use something. Otherwise you feel lot of pressures. So even a pressures of 20 to 30 feet of water we cannot bear when we are having bones, much more stronger material as compared to the microorganism, then also we cannot bear this. While these organisms, those who are known as the barophilic organisms, they are much resistant to these pressures. Now you cannot think. Suppose if I tell you, in terms of, we will discuss the criteria, microbiological criteria or physical criteria later on. Before this, suppose you may be the strongest sumo pelvan, and if you take weight on your head, now that will give the pressures. How many bogie of a railway train you can take on your head until you will feel comfortable? Not even 1/4 bogie. Maximum you will be comfortable having 2 or 3 times of your body weight. Suppose you are 70 kg, then 140 kg or at the most 200, 300 kg, if you take on your head, you may be comfortable. But this barophilic organisms, if you put nearly 100 bogie upside down, still barophilic organism, they may not be crushed. So here if we look to this, barophilic organisms, now as we are preparing for NET, so I have found out the specific terminology which is used. Barophile is normally used, but in NET exam they will find out, or they will use some uncommon terminology, which is known as a piezophilic, or that is piezophilic. It's also known as barophilic. Is an organism which thrive at high pressures, such as a deep sea, which could be bacteria or could be archaea, and they are not normally found at the floor of the sea. So can you think what could be the depth of the sea? If you go to a not seashore, it may be 10 meters, 30 meters, but if you go inside, then